Today, we're breaking down a new lung cancer paper using patient-derived tumouroids to predict treatment response and personalized CAR T-cell therapy in solid tumours, including why immunotherapy fails, how resistance emerges, and what this means for precision oncology. I'm Dr. Susan Heavey, Associate Professor at UCL, and as usual, I will take you through each figure panel by panel first, and then zoom out for a bit of a bigger picture view. Today, that'll be in the form of some wider context from recent literature, then a data viz spotlight on a really cool graph type, and then bigger picture on some clinical relevance as well. You can skip around the video to use it how you like. And for now, let's just jump right into figure 1. So firstly, figure 1 introduces the patient-specific tumouroid platform used in the study. Panel A outlines the workflow. Tumour and matched healthy lung tissue are used to generate tumouroids and organoids, which are then molecularly characterized and tested against therapies, including CAR T cells. In panel B, targeted DNA sequencing shows that tumouroids retain the key mutations present in their parental tumours, while healthy organoids lack cancer-associated alterations. Panel C compares variant allele frequencies between tumours and tumouroids, demonstrating strong correlation and preservation of intratumoral heterogeneity over time. Panel D tracks individual mutations longitudinally, showing genomic stability during extended culture. Finally, Panel E shows DNA methylation profiling, where tumouroids cluster with their parental tumours and remain distinct from healthy tissue, confirming preservation of epigenetic identity. Together, this figure establishes lung tumouroids as a faithful, patient-specific model for therapeutic testing. Figure 2 shows that lung tumouroids preserve the histological and proteogenomic identity of their parental tumours. In panel A, immunohistochemistry demonstrates that key lung cancer markers are retained in tumouroids, closely matching the staining patterns seen in the original tumour tissue. Panel B quantifies this using H-scores, confirming strong concordance between tumours and matched tumouroids across multiple patients. In Panel C, proteomic profiling reveals substantial overlap between tumours and tumouroids, while healthy organoids show a distinct protein signature. Panel D uses dimensionality reduction to show that tumouroids cluster with their parental tumours rather than healthy tissue, reflecting preserved molecular identity. Panel E through H extend this analysis, showing that tumour-specific pathways and mutation-associated proteomic signatures are maintained in tumouroids. Together, this figure confirms that lung tumouroids are not just genetically faithful, but also histologically and proteomically representative of individual patient tumours. Super nice work here. Figure 3 shows that lung tumouroids can model individual patient responses to therapy. In Panel A, live cell imaging tracks tumouroid viability following treatment, visually distinguishing sensitive and resistant responses over time. Panel B quantifies these effects, showing dose-response curves and IC50 values for standard chemotherapies, directly comparing tumouroids with reference lung cancer cell lines. In Panel C and D, experimental responses are normalized and aligned with large pharmacogenomic datasets, demonstrating that tumouroid drug sensitivity mirrors each patient's clinical outcome. Panel E links these functional responses to the proteome, identifying proteins associated with resistance to platinum-based chemotherapy. Together, this figure shows that lung tumouroids do not just resemble patient tumours biologically, but can also predict patient-specific treatment responses. This is the dream. So moving on from those sort of proof of usefulness figures, figure 4 then introduces the patient-specific individualized CAR T-cell selection platform. Panel A outlines the pipeline. Tumour tissue, healthy tissue, and matched organoids are first screened for tumour-associated antigens, which then guide CAR T-cell design and functional testing. In Panel B, immunohistochemistry shows that antigen expression is largely conserved between primary tissues and their matched tumouroids and organoids. Panel C quantifies this across patients, highlighting tumour-specific expression patterns that nominate candidate CAR targets, such as HER2 or PD-L1, while excluding antigens with poor tumour cell activity. Panel D shows the generation of second-generation CAR T cells targeting these antigens. Panels E and F demonstrate antigen-specific CAR T-cell activation and cytokine production when co-cultured with tumouroids, confirming functional recognition. Together, this figure establishes a rational, patient-specific framework for selecting and testing CAR T-cell targets in lung cancer. This is the conceptual centerpiece of the paper. So, Figure 5 tests whether CAR T-cell activation actually translates into tumour killing. Panel A outlines the killing assay, where CAR T cells are co-cultured with tumour and healthy organoids at defined effector-to-target ratios. In panels B and C, live cell imaging shows CAR T cell-mediated killing over time, with clear differences between the tumour and healthy organoids. Panel D quantifies this effect, revealing strong patient-specific variability. Some tumouroids show robust selective killing, while others are resistant, despite similar CAR T cell activation. Panel E profiles cytokine release during killing, showing that activation-associated cytokines do not necessarily predict cytotoxic efficacy. Panels F through I explore underlying mechanisms, including antigen density and immune evasion pathways, highlighting why some tumours resist CAR T-cell killing. Together, this figure demonstrates that effective CAR T-cell therapy depends not just on target recognition, but on tumour intrinsic resistance programs, captured here by patient-derived tumouroids. This delivers the central insight of the paper. This lung tumouroid paper sits in a fast-moving lineage of 3D patient proximal models designed to predict treatment response, but with a twist. The earliest clinical-facing wins came from patient-derived organoids in metastatic colorectal cancer. In the tumouroid study, metastatic lesion organoids were used as an ex vivo chemosensitivity test, showing strong predictive value for some regions, but not all, highlighting that prediction can be treatment-specific. A parallel branch tackled radiotherapy, where rectal cancer organoids were embedded into co-clinical trial designs and shown to predict chemoradiation response with high reported accuracy, a key milestone because radiation response is notoriously hard to forecast from genomics alone. More recently, groups have tried to standardize predictive testing across many therapies, not just one drug class. A unified framework for metastatic colorectal cancer expanded organoid testing to chemotherapy, biologics, and targeted therapies, and explicitly framed feasibility around clinical reporting timelines. In lung cancer, speed has been the bottleneck. One approach used lung cancer organoids on microwell arrays to deliver drug response readouts within about a week, aiming to make turnaround times clinically actionable. And then there's the microenvironment problem. Organoids are powerful, but often lose intact tumour architecture and native immune context, which is why patient-derived explants and tumour slice cultures keep resurfacing for immunotherapy testing, because they preserve more of the original tissue ecosystem. What's distinctive here is the precision immunotherapy focus with safety built in. The platform pairs tumour and matched healthy organoids, then uses functional CAR T assays to assess both efficacy and on-target off-tumour risk in a patient-specific way. Moving beyond prediction for chemo alone into engineered T-cell therapy selective. In short, organoids have proven that they can predict, but this paper is part of the next wave asking whether they can predict immunotherapy performance and toxicity, not just drug sensitivity. Okay, so let's use figure 3F from this paper to do a little data viz spotlight. So this panel uses paired volcano plots to summarize proteomics data, and it's doing a lot of conceptual work in a very compact space. Each dot here represents a single protein quantified by mass spectrometry. The x-axis shows the log base 2 fold change, in other words, how much the abundance of that protein differs between two conditions. Proteins to the right are higher in tumour or tumouroid samples. Proteins to the left are higher in healthy or organoids. The y-axis shows minus log base 10 of the adjusted p-value. So this is a way of visualizing statistical confidence. The higher a protein appears on the plot, the more confident we are that the observed difference is real and not noise. Because these two dimensions, effect size and confidence, are shown together, proteins that are both strongly changed and statistically significant rise into the upper corners of the plot, creating the characteristic volcano shape. The color coding here is important. Gray points are not significant. Green points pass the adjusted p-value threshold. Yellow points pass both the statistical threshold and a predefined fold change threshold. This tells us the authors are explicitly filtering for changes that are not just statistically detectable, but also biologically meaningful, at least according to the thresholds that they can set. And this is good standard practice. What makes this panel especially powerful is that the same analysis is shown twice. The top plot compares tumour tissue to healthy tissue, while the bottom plot compares tumouroids to healthy organoids. This paired design allows a visual question that would be much harder to answer from tables alone. Do tumouroids reproduce the same tumour-associated proteomic shifts we see in patient tissue? And the answer visually is largely yes. Many of the proteins that are strongly and significantly altered in tumour tissue show similar directional changes in tumouroids. That visual concordance supports the author's claim that these models preserve tumour-specific biological programs rather than simply reflecting generic in vitro growth effects. From a teaching perspective, this is also a really clean example of good practice. The authors control for multiple testing, they integrate effect size and significance in a single plot, they label only a subset of biologically relevant proteins to avoid clutter, and they use symmetry between panels to make a conceptual argument visually rather than verbally. For students, PhD candidates and postdocs, this figure is a template worth copying. Not because volcano plots are fashionable, très chic, but because they communicate complex, high-dimensional data in a way that's transparent, defensible, and reviewer-friendly. To understand why this paper matters, it's worth being explicit about the clinical problem it's trying to address. Lung cancer remains the leading cause of cancer-related death worldwide, and around 85% of cases are non-small cell lung cancer. While survival has improved with chemoimmunotherapy, outcomes for patients with advanced disease remain poor. For metastatic non-small cell lung cancer overall, the 5-year survival rate is still under 10% once disease has spread distantly. Even in the modern immunotherapy era, most patients eventually develop resistance and experience progression. First-line treatment for patients without targetable mutations typically involves platinum-based chemotherapy combined with PD-1 or PD-L1 blockade. This has improved median survival, but it's not curative. Real-world data and clinical trials consistently show that the majority of patients relapse. This situation becomes particularly stark after progression on first-line chemoimmunotherapy. There's no clearly effective standard of care in this setting. Second-line options are largely limited to cytotoxic chemotherapy, such as docetaxel alone or combined with anti-angiogenic agents, and these treatments offer modest benefit at best. Response rates are low, progression-free survival is measured in months, and median overall survival after second-line therapy is often under a year. A substantial proportion of patients are too unwell to receive further treatment at all. This is the clinical gap. Patients with advanced lung cancer who have exhausted standard chemoimmunotherapy options have no actionable mutations and face limited, largely non-personalized treatments with poor durability. At the same time, cell-based immunotherapies like CAR T-cells have transformed outcomes in blood cancers, but have so far failed to deliver comparable success in solid tumours. Response rates in solid tumour CAR T trials remain low, and on-target off-tumour toxicity is a major concern. That combination, poor post-immunotherapy outcomes in lung cancer and the difficulty of translating CAR T therapy safely into solid tumours, defines the unmet clinical need this paper is addressing. What this tumouroid platform offers is not a therapy by itself, but a way to rationally test, stratify, and potentially de-risk personalised CAR T strategies before they ever reach the patient, which is exactly what is missing in this part of the treatment landscape. So to bring this all together, this paper shows that lung tumouroids can faithfully preserve tumour biology across multiple levels, from genomics and proteomics through to functional drug response and immune interaction. We've seen that these models don't just look like the original tumours, they behave like them. They reproduce patient-specific responses to standard therapies, they reveal tumour intrinsic resistance programs, and they expose the gap between CAR T activation and actual tumour killing. Clinically, that matters because patients with advanced lung cancer who progress after chemoimmunotherapy have very little options. There's no clear standard beyond modestly effective chemotherapy, and while CAR T therapy has transformed blood cancers, it has struggled in solid tumours because of target selection, resistance, and safety concerns. What this platform offers is a way to integrate all of that before a therapy reaches the patient, to test efficacy, select targets, and flag potential off-tumour risk in a patient-specific way. It's not a treatment in itself, but it's a decision-making framework for precision immunotherapy in a setting where we urgently need better tools. If you're interested in how these kinds of complex human models are being pushed even further by combining multiple cell types to capture tumour immune interactions, I'd really recommend checking out our video on the assembloid paper, where we explore how multi-component 3D systems can extend what organoids and tumouroids alone can do. Thanks for watching, and as always, the paper link is in the description if you'd like to dig into the data yourself, along with some other relevant references.